Well, thanks for the invitation. I was speaking to a distinguished audience about 5 years ago, and I went through my slides, and it's incredible the number of things that have changed. New technologies that we didn't even hear about 5 years ago and now are becoming mainstream and they cover The Economist, Time Magazine, and so on. So I'll tell you a little bit about the story of how we're trying to use genetics and new technologies to help breeding in the UK. And I always start with a picture of the field because when we do research, we do lots of research in the glasshouse, but at the end, it has to be a crop in a farmer's field that then can actually be processed by you guys, sold on to consumers. So the main messages, I'll start from the beginning in case anyone needs to leave. We're in the middle of a DNA revolution as well. So we hear a lot about phenotyping, what we see, but it's incredible the amount of things we can do now with what we can't see, and that's DNA. It's always been there, but now we can see it in ways that we couldn't do just a few months ago. Wheat has a huge hidden potential. I'll tell you why this DNA revolution is allowing us to see wheat in a different light. The question has been posed before, is it sustainable to continue to ignore transgenics? We just ignore it. We've lost the battle. Why fight it? I think we need to have this opportunity to say, and it has to be from you guys, because we're academics, we're all saying, yes, it's a science-driven technology, it's fine, but we need industry to actually step up and say, we want these changes. We want the discussion. We want the discussion, perhaps, and to get there. And last but not least, how many of you have heard about CRISPRs or Cas9? Okay, so this is going to change the world. It's changing the world already. And when I spoke to you 5 years ago, it was in Specialist Magazine, basic science, who cares about this? 5 years later, it's changing the world. And again, that'll be one of my take-homes at the end will be careful what we wish for when we say applied, applied, applied. I'm the most applied person in my institute, but I'm actually on this side, I'm saying basic science is not the enemy of applied science. They go hand in hand, and we need to be sure that continuum stretches, because the discoveries that we make from the basic science at the end many times are the ones that make the breakthroughs in the application, rather than trying to solve the problem here in the field here. So we need to just be mindful that we need to balance both things. So we're in the middle of this DNA revolution because basically the sequencing, the cost of sequencing has gone down incredibly. And not just sequencing, but actually synthesizing DNA. That's another new area, synthetic biology, which I won't discuss. But that's the cost of sequencing a human genome, $100 million. The first one, a few years ago, it was still $10 million. And suddenly, that thing has gone down incredibly. So basically, this is Watson and Crick. And now they probably never imagined that you could actually sequence their DNA for about $1,000. Even with the exchange rate, that's still really, really, really cheap. So you can see that, and you say, well, you know, this technology, and look at the scale. That's not just a normal scale, it's a log scale. So that's actually huge step changes in how it's changing. And suddenly, you know, when we thought it couldn't get better, a couple of months ago, new technology came. We can now sequence it for $1,000. So in a way, perhaps we won't, but all our kids, all our grandchildren will have their DNA sequenced without a doubt within the next 10 years. I've had my DNA sequenced because why? We have these things like 23andMe. So I paid £100, I got my DNA sequenced. I'm probably not all sequenced, but all just the genes. But basically, when it's becoming £1,000, it's just going to be a medical necessity to just sequence your genome. And then how does that offer opportunities to say, well, what does a gene have to do with my industry? Well, when you start looking at what Marks & Spencer is doing, they're saying some of the consumers already know, by doing this kit, they know that they have a certain gene called GSTM and prevents them from using certain chemicals. They know that if they have this broccoli that has higher levels of this, it actually will be beneficial for this group. So people are data-driven. As soon as they can get more information about themselves, they will, when the costs go down to where they are now. And they'll start asking, well, I know I have this gene. I want to know, I want to eat things that will make me feel better, and so on. And of course, it's not just for humans. You can imagine cattle. If they have certain genes that makes them process food in a certain way, if we know that, then the feedstock will change it, and so on and so forth. The same thing with plants, right? So having this ability to sequence DNA just opens up new opportunities, and I think that, you know, as industry, you should think about how this DNA revolution will change the way you think about your problems and how you can address them. We've been able to sequence many crop genomes in the last 5 years, so tomatoes and maize and so on and so forth. And just recently, we were able to complete the genome sequence of wheat. And that's a huge landmark because wheat is very, very complicated, because wheat is what we call a polyploid. It has many ploids, it has many copies of every gene. So actually, wheat is a hybrid between 3 species. Basically, these 3 little grasses that you probably wouldn't even care to look at them, they actually make up 20% of the calories and 25% of the protein that the world eats. And they came together to form wheat. okay? And it's very important because wheat means that every gene in wheat, and this little circle represents a gene in wheat, every wheat plant has 3 copies of every gene, and I'll tell you why that's so important later on. So it's been very hard to sequence the wheat genome because, first of all, you have 3 copies of every gene. These things are very, very similar. So it's like having a huge jigsaw puzzle. The pieces are 98% similar, and they're basically repetitive. So it's been really, really hard. And when you start putting, this is the DNA. Basically, when we talk about DNA, this is DNA, A, C, T's, and G's. There's a Word document. If you put them on a Word document and you make very small margins, very small font, it's basically the wheat genome has 4 million pages of Word of DNA sequence. We're pretty simple. We guys have about 1 million pages. Wheat has 4 million pages of DNA. So when we're trying to understand it, it's been very hard until now because we try to understand what are the genes, what is this DNA that's doing. But first of all, even just to look at 4 million pages has been very hard. And just recently, we've got the first glimpse of them. So now we have the complete puzzle. We have the 4 million bases, or the 4 million pages. And now we can say, how do we actually look at it and examine it to get that information? So with this sequencing technology, it doesn't just mean that we can look at the wheat plant. We can also start looking how the wheat plant interacts with its environment and basically its predators, or with microbes, in this case, with yellow rust. So this is a nice picture of a yellow rust spores just literally breaking through the leaf and coming out. And basically, this technology allows us to go into a very complicated sample. That could be roots. It could be the rumen of cows. It could be a leaf infected with rust. Sequence that and understand what the pathogen is, how the plant is responding, and how they're interacting. And so, again, that question, we could just not even imagine about doing it just a few years ago. And there's a project that basically we started with Diane. So Diane's been leading this project. And in this case, we have the institutes, but we also have a lot of the companies, both breeders and agronomy companies, thinking about how we can use this technology. And one way was that we started sequencing from the field. So now, not instead of doing these trials in the glass house, but going to the field, farmers' fields, taking leaves, and then seeing what yellow rust was in those fields. And we started seeing there's different types of yellow rust in the field. Those different yellow rust respond differently to different chemicals. So that's why it's the interest you have of Syngenta and BASF. And then when we start looking at the pattern of when these yellow rust appear in the field, we see that depending on the time of winter, spring, or summer, different types of yellow rust appear. So you can imagine that having your variety, and if you know your variety is susceptible to this isolate, you have to be very careful in May and so on. So we can now start seeing, instead of having one static snapshot, we can see actually throughout the year which yellow rust is there, how varieties respond to it, at a level that, again, it wasn't possible before. And you can imagine now putting this together with the maps that we just saw, you can start predicting, well, I know I have farmers' fields that have this variety, how will this affect and how will pathogens spread? So, can I ask that you start the video, please, in the back? Thanks. So, another problem that faces industry from breeders and farmers is this issue of pre-harvest sprouting, which is germination of wheat lines. So these are lines that these are wheat plants that have been collected from the field, harvested just at the time they're supposed to be harvested. This line here and this line here are 2 UK varieties. They're almost identical except for one gene. And that one gene makes them resistant. You can see here. This is basically 6 days of an image, just seeing how this has completely germinated or sprouted before its time. These plants here basically are identical for one gene. And basically, those sprout, but those sprout 10 days later. So basically, when you have a late rain and you can't harvest, you lose your crop, you're still fine, and they will still germinate, of course, when you sow them 1 month later. So we were able to work with breeders. Again, there's a project funded by HDB, and with the breeders to see, can we find that gene? And what does that enable us to do? It enables us to, instead of having to look at those 4 million pages of DNA, we can make a very simple question once we find the gene. If you have a DNA marker over here or here, you can tell if the plant is going to be resistant or susceptible. So instead of having to do a huge effort on trying to sample the plants in the field, you just collect DNA, you run a marker, a genetic marker, and if it's an orange, it's susceptible. If it's this color here, it's resistant. So now breeders, instead of trying to select varieties from here, they can just discard this. They can go here. And since breeding is a numbers game, it skews the numbers in your favor when you can select for this. And you can imagine for many other traits, we're now developing markers or perfect markers that actually tag the traits that are of relevance for industry. What we can do next is say, okay, we have this big, basically, pedigree of all the UK varieties. We can see which lines have the susceptible or resistant. And we actually see that breeders have now started to select for all the UK varieties for group 1, which are the bread-making varieties. All of them have introduced now the resistant version of this gene. compared to the bread making or the feed varieties that basically are not, the Hagenbeck falling number of pre-harvest sprouting is not as important. But basically by doing this, they can actually breed varieties that have the gene and are basically less prone to pre-harvest sprouting. And of course, whole issues of sustainability and so on there. And of course, we can now understand the pedigrees, and now they're generating crosses based on specific varieties and following these genes and so on. There's one example, but these are now many, many genes that we are now, for the first time, being able to uncover that we couldn't do before. One of the other traits we're looking at is trying to understand yield. So we have yield. Yield is determined by how long and how wide each grain is. So after a lot of work, we've been able to find some genes that affect yield. Okay, so great. And basically, these are just like the plants I showed you that were susceptible and resistant to sprouting. These are lines that have high yield and low yield. So these are the two grains. You're probably trying to squint and see, is there any difference? I'll show you different. That's the big grain. That's the small grain. So thankfully, the funders are not here, because basically they're funding us to identify that. And you say, yeah, what the hell am I doing? But basically, when you look at that, that's 5%. If you put 20 grains together, that's basically one extra grain at the end there. In one spike, that's 2 extra grains. In one field, it's 500 extra loaves of bread. So actually, that is what breeders are trying to select. This, very, very small, is the 5% yield. So the question is, we're trying to get to the gene to understand it, but now the critical part of wheat is that we have a huge potential that we haven't tapped. And I'll explain why. So a lot of these genes that we've seen are genes that actually put brakes. They're actually genes that stop the plant from growing. They stop the grain from growing. And wheat, of course, has 3 copies of every gene. So basically, wheat, we have the 3 genomes come together, and basically this wheat plant will have these 3 copies. In normal, let's say normal, like a diploid plant, like maize or rice or barley, they only have 1 copy of every gene. So if you imagine here, if I had a master switch and I could just turn the lights on and off, basically that's what maize does. It can mutate that gene, and if it mutates, you have the lights on or off. You have the gene working or not working. In wheat, on the other hand, you can do the same thing with 1 gene, but the other 2 genes still work. So breeders for a long time can't see variation like they can see in barley, in maize, and rice. And there you see huge variation. In wheat, very, very subtle, because you have 3 genes versus 2 genes. You have 3 breaks versus 2. It's very small effects, very subtle effects, and that's 5% yield. So now the intriguing question comes, what happens if we go from that to that? That's 5% yield. What happens if we do that? And that's something that's never happened before, because the chances of that happening is like winning the lottery today, right again, and again in the same shop, the same person, with the same numbers almost. It just never happened. Breeders have had no chance to look at that plant. They've been playing with that. And now all of a sudden, by having the genome, we can actually do that. And that really changes the game of how we do it. And it's not just by chance, it's by purpose. And that's the big difference. So you're probably thinking again, am I crazy? What are these 2 things? What do they have in common? Okay, and it's a huge consequence for anyone who works with plants, because a lot of the funding that we've received in plants has gone for what we call this basic science, Arabidopsis. That's Arabidopsis. That's a weed that you probably kill many times a day when you walk. It's just a weed that grows everywhere. But it's great to study it because it's very simple, it's fast to grow, and so on. That's a platypus. So basically, a wheat plant is to Arabidopsis like we are to a platypus. So we have to be very careful when we make these conclusions about Arabidopsis being the model plant. It's like we chose the platypus to be the model animal. We wouldn't be happy with it. And I think now, in a way, we hadn't had the tools, we didn't have the opportunity to work with these, but this DNA revolution has changed that. Now, for the first time, you can start saying, well, I'm not happy with this model for us. We can actually start doing it in the crop. And we can start doing some fascinating science, which is important for career development for the scientists to actually be able to do high-level science in the crop, but also have an immediate impact in the field. And that, again, changes the game. So one of the things that we've done is something very simple. We've taken a big bag of seed. We've put a chemical in it that mutagenizes it. This is something that breeders have done for many, many, many years. Golden Promise, many barley varieties are coming from mutagenesis. But then the critical thing is that now we can sequence them. So we actually taken this big collection of plants, we sequenced it, and now we know all the bases, and this plant has a mutation in this specific position. And now, since we have all the 4 million pages, and we know this plant has a mutation, this plant here might have a different function. And this allows us to ask this question. If I want to make a mutation in all the 3 copies, I can now do it because I can go to a database, find those 3 plants, and order them, and within 20 minutes, I can start making my experiment, whereas before, that would have taken me a whole PhD to just identify those mutants. Now it takes 5 minutes, and that's a game changer. And just to give you an idea, so since we first released this, it's still not public, because we're still finishing it. Breeders have ordered more than 800 different mutants to start putting into UK varieties. This is done in cadenza, which is a UK wheat. Okay, so basically these mutations are now out there, and they're going to start getting into the food chain very soon. So we can do mutagenesis as one way, but then again, is it sustainable to continue to ignore transgenics? You've probably all seen Purple Tomatoes, work by Kathy Martin. And again, most people don't want to take a life extension pill. They prefer to eat a nice, lovely tomato, and they prefer to eat a tomato for 30p than to have to buy the blueberry or the acai berries from Brazil to get that energy or to get those antioxidants. And of course, this also has health properties, and it also has extended shelf life. Work by Jonathan Jones trying to make potato blight resistant varieties. From this wild relative, he's found several genes that give it resistance. So basically, when he puts it into a plant transgenically, it's resistant. You can then put it into field trials. No chemical at all, and that plant is beautiful. That's in the UK. So you can imagine how all of a sudden now we can use these transgenic technologies, but we're just blind to it because, well, we can't really use them. Is it sustainable to get fish from the ocean, make it into a fish meal, to give it to fish? Is that sustainable, really? Or the work that Jonathan Neighbor's done at Rothamsted to find genes in algae, put them into brassica, and now brassica that produces fatty acids with the same profile as fish that can be given to fish. Much better to give brassica oil to fish than to give fish meal to fish. We know what happened with prions, with mad cow disease, so it's just not sustainable. And I think we need to make that argument out. And I think these are 3 beautiful examples of less pesticide, more sustainable farming practices, and Kathy's work with antioxidants that we should herald as new ways of doing technology or using biotechnology. The last example I want to show is these new plant breeding technologies, CRISPR or Cas9. So if you haven't heard of it, you will hear about it, and if you keep your eye open, you'll see it. So basically, these transgenics that we've been using until now are basically quite, in a way, they are quite messy. That's why we have regulation, and that's why regulation is good. It's not blocking it, but regulating it, because these can go anywhere, and basically they have usually a lot of baggage with them. These new technologies, basically, we can actually modify plant genomes without inserting any foreign DNA. And the critical thing is that in many cases, they leave no trace. And that's the big issue. How do you regulate something that could have happened in nature that there's no trace of it? You don't know. There's no way to tell the difference. Okay? And basically, this Cas9 technology is a way to precisely modify the genome. And basically, you can make modifications that you cannot tell them apart from the chemical that I used before, or from the mutations that we're receiving being exposed by the sunlight. It just happens every day. We're all mutating. But the problem is that now we have a way of doing it precisely, and there's no way to tell that it was done that way. So it just changes how the regulation has to work, and that's why Europe has been so muddled with it, because they want to regulate it, but you can't. It's the same. So how do you regulate something that is the same? So basically, I could show you, this is one way of showing it to you. That's another way. But basically, the take-home is that this CRISPR Cas9 is just a big pair of scissors. And the pair of scissors becomes very powerful because now you have a pair of scissors, and you have the whole genome. And this pair of scissors, depending on how you make them, actually, you can target it to one specific region of the DNA. So again, those 4 million pages of DNA sequence we had, we can actually target it to one specific region of that whole DNA. And that, again, changes how we do it. And this just to show you a little schematic in The Economist, very mainstream, you can see. Basically, you have DNA. You have this Cas9 enzyme. And basically, by having the guide will take the scissors, will find exactly in the DNA where you want to cut, and basically will then cut that and basically either not introduce DNA or put new DNA or fix the DNA, and there's many, many applications. Originally, it was done just to cut, and when the plant repairs itself or the humans repair ourselves, we make mistakes, and that changes the DNA. Now, new technology basically can actually put new DNA or fix DNA. And now this is really coming online now, because you can imagine that the cover of The Economist, the cover of National Geographic in August of this year was basically the DNA revolution with this gene editing, improving humans with this gene editing. And again, even in Wired magazine, this genome editing technology is going to change how we do it. And now the value of GM or gene editing technologies will probably become much more apparent to the general public, because basically, if you can fix a disease, everyone's happy to use technology. Right? So everyone's happy with that. And now, basically, the prospect of improving human health with this technology will come, and it's coming now very fast. This technology, we didn't know about it 5 years ago in the mainstream. Now it's completely mainstream, as you can see here. So now when that opportunity arises, I think we should be ready as a group to say, well, this technology also has really advantages for agriculture and for processing that we should take advantage of. So just to be mindful that this is the August issue of the National Geographic and August from The Economist, if you want to have a look, because it's really good reading both of them. And just to give you an idea of how we're using this specifically in the UK, this is work that we've done with Lars Ostergaard, pod shattering in oilseed rape, big problem. By understanding from the model species Arabidopsis that looks like brassica, we know that if we basically take a gene out, we make a plant that doesn't shatter, and basically we find a gene, we can make a mutation in the gene, and basically the pod, which is here, where it actually shatters, where it actually breaks, we basically seal it. It doesn't break anymore. So pod shattering is not there. The problem is that it seals too well, and you have to have like a hammer to open it. So now the question is that can we regulate that in a slightly different way? And that's a big project that Lars is leading as part of the Crop Improvement Club to get that done. Okay, so just the final thought, we are in this DNA revolution. I think it's important that you're aware and that you see that these costs are just changing how we answer problems and the questions we can ask. And you should think about how you can think about or how you can implement DNA technologies in your fields and what are the consequences of that. The huge potential of wheat that I think we're going to just starting to tap the surface of. Is it sustainable to continue to ignore transgenics? And again, this Cas9, this CRISPR-Cas9 technology, which is kind of the technology that's coming now, it's literally going to change the game. And again, we need to be sure that we're thinking of addressing that as well. From our point of view, from the academics, we really do need industry to be able to get this potential to the public. So that's why we're here and we are really interested. Many times we don't know exactly your needs, but I think that we need to find ways to have that communication. The sense of urgency is important because even though we have the best intentions, it still takes time to take these discoveries into the field. So that's something important to have in mind, that when we think about funding and so on, you need to be thinking what's in the future, because if you have a problem today, it might be hard to have a solution today, but if you say in the next 10 years we need to be there, we can probably step up, especially with genetics, which is a little bit more long-term, as you were saying before about generation times. Again, the big point, be careful what you wish for. Basic science is not the enemy of applied science. They're one and the same, they're a continuum, and we should foster both. Because if we just go for the applied science, we might not be getting the best science that we want, and we might be actually cutting out the ideas that will help us in the next 10 years. So we need to be careful, and we should actually be asking for more science rather than more applied science and less basic science. So just be mindful of that. And then, again, as pointed out before, it's not just about genetic diversity, but also human diversity. Thank you very much.
Thank you.